So just wanted to give a brief introduction about oligonucleotides. So they do work through multiple mechanisms, including modulation of RNA function or binding to protein targets to produce a pharmacological effect. These can be very diverse structures, as short strands of chemically modified nucleotides that increase the stability of these within the plasma and get to the proper target. So we work on both antisense oligonucleotides as well as siRNAs. And we have been focusing on this for liver diseases, and primarily what I'll be talking about today is for hepatitis B. So preclinical toxicity of oligos and the translation to the clinic, the most common target organs there are the liver and the kidney, primarily because that's the two organs that see the most of the oligonucleotides in the body, and then followed by the spleen. So there's examples of translation of this to the clinic. There's 2 previously approved ASOs that have warning label on it and that have to have a REMS program to monitor for that. So what we do see is that you can have accumulation, in the liver and kidney. The endosomes can actually accumulate these oligos, and they have these sort of basophilic granules in the Kupffer cells in the liver and in proximal tubules, and this is known as a stippling effect. So this is generally considered non-adverse. It's just related to the oligo uptake and processing, and doesn't have any effects on function or damage. But of course, as you start pumping more and more of these oligos in, they can certainly have long-term effects. One of the other things that we do sometimes see, mostly with the older generation oligonucleotides, is that you can have some alterations in hematological and coagulation parameters. Those have been observed both preclinically and then have shown effects in the clinic as well. One of these is an ASO, particularly where they had to monitor for platelets in the clinic. What we do sometimes see in the rodent particularly is these pro-inflammatory effects. And those are, it's likely due to off-target stimulation of the immune system. It's generally not something that has been translated to the clinic, and it is something that we have to manage in our non-clinical studies. So taking a step back here to talk a little bit about our approaches to curing hepatitis B. So similar to hepatitis C, where you really need to attack the virus on multiple different pathways. So one is to inhibit viral replication. So we have a small molecule capsid assembly modulator. Also then lowering the antigen burden, and that's what I'll be talking about today, the focus. So decreasing the amount of secreted antigen. And we can do that either through siRNAs or through ASOs that are targeted to the HBV virus. And then the third approach is to boost the immune response. And so what we're doing here is another small molecule program that's targeting PD-L1. That will actually go to the liver. So what we are trying to achieve is to actually have a functional cure by combining multiple of these kinds of mechanisms and ultimately eradicate the HBV virus. So I'll talk first about our siRNAs. So we have it's ALG 755. It has a GalNAc on one end of the siRNA, and this allows the active uptake of the siRNA into the cell through the ASGP receptors that are on hepatocytes. We've selected this particular sequence because it has almost no mismatches to any of the different HBV clinical isolates. So we believe that that's going to be an effective way of treating HBV. We also have some proprietary stabilization chemistries within this siRNA, so that it will be maintained in the plasma and have the effects that we're looking for. So one of our workhorse models is a AAV HBV mouse model. There's another siRNA that's currently in clinical trials right now, this VIR-2218. If you dose the siRNA every 2 weeks, you can see that you reduce the S antigen here effectively. Once you stop treating with the siRNA, we see that they go back to virtual baseline values. With our 755 siRNA in the same model, we have a much more sustained response. And even after discontinuation, you still continue to see the decreased S antigen levels. So what we've used for this model is to create a PK/PD correlation in the mice. And from our experience with this particular model and also looking at clinically validated molecules, what we found is that the C average appears to be the most relevant PK parameter. When we built this model, this allowed us to help predict what the efficacious dose and exposure was going to be. So looking at some of the PK properties in non-clinical species, so for this siRNA work, we have used both rat and monkey as our 2 species. Because this is targeting HBV virus, essentially what you're looking at is any potential off-target effects there. So you can see that in both the rat and the monkey, the plasma exposures are very short-lived, but you have a very long tissue half-life, particularly looking at the liver, which is where we want the siRNA to be. So one of the things that is particularly problematic for oligos is trying to understand any off-target effects. So we do pretty deep dive into the bioinformatics and try to identify any potential off-target genes that may have 2 or 3 mismatches and that could then have effects particularly in the liver and the kidney. So we did have a couple of those. So you can see here that we only identified about 7 genes and they had 4 mismatches there. So then what we did is to actually go look to see if we had any effects of gene expression, looking specifically targeted at those genes that we had identified from an in silico perspective, and actually tested that in vitro and found that indeed they did not cause any change in gene expression. What we've also started to do, and I think many others in the industry, is looking at RNA-seq, so looking at the HepG2 cell line. So we looked at the concentration at 10 nanomolar, which is essentially 1,000-fold above the EC50 against the HBV virus. And what we saw was that there was very few genes that were actually decreased even at these super therapeutic concentrations. So this was all good news for us. And so we went into our GLP tox studies. One thing that we always monitor is looking at a number of different donors, human PBMCs, to see if we had any immune stimulation. And in fact, we didn't have anything there. Negative in the in vitro gene tox assays, negative up to a maximum feasible dose of 2,000 mg per kg in the rat micronucleus test. We had no changes in hemodynamics, ECGs, or respiratory parameters, no neurobehavioral effects up to the highest dose that we tested there, which was 100 mg per kg. When we went in to do our general tox studies, we did a 5-week 6-dose, so giving a dose of the siRNA every week subcutaneously. We dosed vehicle 10, 30, and 100 mg per kg in both those species. And then we looked at recovery for 4 weeks there. In fact, that high dose that we tested at 100 mg per kg was our NOEL in both the rat and in the monkey. So this overall gave us a favorable non-clinical profile and allowed us to advance. So one of the things that we had seen before from another competitor was this Alnylam HBV. So this was another siRNA that was targeting HBV. And what they saw was even after a single dose in humans that they were having ALT elevations. Vir actually made one base pair modification to this, and that alleviated the toxicity, and they were able to advance this into the clinic, which is currently being tested. So what we did is we took, this was in collaboration with Phoenix Bio in Japan. So these mice have 85 to 90% of their liver is actually humanized. So we really wanted to test and see if this kind of model would actually predict the toxicity that the Alnylam had shown. So even after a single dose, you can see we have these elevations of ALT. And then after you give repeated dosing, it continues to go up. The VIR-2218 compound, by comparison, shows a much lesser effect on ALT. And then for our compound, the ALG-755, it was virtually indistinguishable from the vehicle control. So we were able to use this kind of model to confirm that the other competitor molecule actually was hepatotoxic in this model. It's not hepatotoxic in normal rodents. So this was a good model to be able to predict that toxicity and to give us some reassurance that with our 755 molecule that we were not going to have hepatotoxicity in the clinic. So to summarize this portion, siRNAs have certainly demonstrated the clinical validation in HBV-infected patients. We designed our siRNA sequences using proprietary technology and have liver targeting to maximize both the in vitro and the in vivo potency. The proprietary chemistries actually increased the potency of the siRNA and the stability as well. We have an exclusive license to the GalNAc technology that is used to target into the liver. So we filed a CTA last August, and then we initiated dosing in healthy volunteers, and we've just started to dose hepatitis B patients at the end of last year. So far, we have not had any safety signals, and we've been able to escalate the dose in the clinic. So fingers crossed that we'll have some good news coming out from the CHB patients soon. So switching gears a little bit here, we had also had an approach to look at antisense oligos for that. ASOs, have been in the market. They were actually in the market before siRNAs, but there have been a lot of failures and withdrawals in the clinic, mostly due to safety liabilities. The mechanism of action of ASOs is different than the siRNAs, but it also reduces this surface antigen secretion just as the siRNAs do. We wanted to go after both approaches here. So the duration of the action differs between the two modalities. So ASOs have a much faster onset to effect, but once you stop treating, it's also a faster return back to baseline. ASOs in the clinic likely need more frequent administration than siRNAs, but it may be a complementary approach as well. So we identified a lead candidate, this 572, that it potently inhibits the secretion of HBs antigen in vitro. We also did the same sort of off-target analysis by using in silico methods, and then also did the same sort of testing in vitro in human cells, and we didn't identify any genes that were downregulated in response to the treatment of the ASO. So again, going back to our workhorse model, the AAV HBV mouse model, and here we have to dose much more frequently. So we give a loading dose twice weekly for 2 weeks, and then it's a weekly maintenance dose thereafter. So if we look at the 572 at 1 and 3 mg per kg, which is ballpark of where we think the human efficacious dose is going to be. They show similar effects. If you dose even higher, up to this 10 mg per kg, you get even more efficacious decrease of S antigen release. Similar to the siRNA, we also created another PK/PD correlation. What we saw was that the concentration required in the liver is actually higher relative for an ASO than for the siRNA. So we do believe that we would have to dose more frequently in the clinic. Looking at both the single dose PK in the mouse and the monkey, also similar to siRNAs, where we have much higher exposures in the liver than we do in the plasma. It's very short-lived. When we have the GalNAc conjugation on here, we also see very rapid absorption and distribution and a very short plasma half-life. We have good systemic bioavailability, and it mostly is showing up in the liver and the kidney, as we would expect. We also did some experiments where we dosed the ASO that was not conjugated, and also saw that we had similar exposures in the liver and kidney, but not as high as the GalNAc conjugation. So clearly that was working to get it into the liver. So similar to the siRNAs, we had no immune stimulation, no gene tox signals, no safety pharmacology signals in mice or monkeys. It was very well tolerated, so we did the same sort of dosing regimen that we were going to be testing in the clinic. So giving a loading dose twice weekly for 2 weeks and then weekly thereafter. We dosed here because we were dosing twice a week. We only went up to about a 50 mg per kg dose level. We saw no safety signals there, so both the mouse and the monkey, the NOAELs were the highest dose tested. So we had no safety signals identified in our non-clinical studies or in the single ascending dose in healthy volunteers up to a 480 milligram dose level. So then we backed down on the dose and we dosed in patients at 210 milligrams, again following this same dosing regimen. This is where we had a big surprise. We had very little efficacy, even at this dose level. And unfortunately, 4 of the 6 active subjects had elevations of ALT. Very concerning for DILI, particularly when you're in a population that's already susceptible to liver effects. I can tell you we all had a lot of sleepless nights after this. We had one subject actually went up to as high as 3,000 his ALT levels, and because these are have such a long residence time in the tissue, it took a long time for that patient to actually come back down to normal ALT levels. Now, of course, we have a ALT flare committee that's reviewing all of this data because that's part of the measurement for efficacy. And we couldn't find any reason that this would be related to having an ALT flare that's part of the cure, it really did seem to be purely a drug-induced liver injury signal. So we discontinued dosing permanently in March of last year, and we decided to close the program because there was such a level of toxicity there and very limited antiviral activity. And we really couldn't understand where this had come from. I mean, it was not something that happened in healthy volunteers. It was really limited to patients with HBV. So there's some kind of interplay happening there between having the virus and presumably having these off-target effects. So because we didn't have this translation, we were trying to think, how could we have predicted this? What could we have done differently? Which many times is the way that we make big advancements by, excuse me, by trying to understand where things have gone wrong. So we had a collaboration with Inspiro, which is a company that makes these 3D spheroids. And we decided that we wanted to test our ASO, but we also wanted to look at other competitors' molecules. So we selected a number of ASOs based on either the published data or the experimental data that we had generated in-house. We took some ASOs that had shown signs of hepatotoxicity in either a preclinical species or in humans. And then we also had some others that didn't have any hepatotoxicity. So we made all of these ASOs, we formulated them, and sent them only ALG numbers so that they were blinded as to what they were actually treating the spheroids with. So we take these human liver microtissues, and they were maintained for 14 days. We tested a top concentration of 100 micromolar and then did half-log dilutions down. We replaced the media on 3 separate occasions and always dosed with fresh ASO every time that we were replacing the media. So we collected the media here and we looked at LDH release. In the future, I think they've gotten to the point where they can actually measure ALT from these cells, even though it's only like 1,000 cells. And then at the end of the 14-day treatment, we looked at intracellular ATP levels by cell titer glow. So this was a joint agreement between the 2 companies. Obviously, they wanted to understand if their model could predict some of these hepatotoxicities. So it was a good opportunity for both of us. So I won't go too much into this, but we had a number of different ASOs that have gone into the clinic where we had either in vivo toxicity or clinical effects. So we had a mix of positive and negative control. And then also took our 572 as well as the parent ASO of that without the GalNAc conjugation. And what we saw, so this was the EZN and an Ionis molecule that have both shown toxicity in the clinic, that we saw clear effects where we were having decrease in ATP levels. And also you can see that we had tremendous LDH release over time by treating there. And then looking at some of the negative controls, we didn't have any kind of toxicity observed and no real LDH release either. Now, interestingly, when we looked at our molecule, surprisingly, the 572 that had the GalNAc conjugation really didn't show any cytotoxicity, but the parent ASO, which presumably should be harder to get into the hepatocytes and cause the damage because it doesn't have this GalNAc conjugation, actually showed some toxicity there. One of the other things that we had done was to take a molecule that had toxicity and we made some chemical modifications there. which seem to reduce the in vitro cytotoxicity profile. And we showed that with these kind of modifications, we could actually reduce the toxicity by about 5-fold in that case. So overall, it looked as though based on this limited data set, that we actually had a pretty good correlation between known hepatotoxicants and actually causing cytotoxicity in the spheroids. So there's generally pretty good concordance there between the LDH release as well as the ATP levels. Generally, if you had a measurable IC50 in this assay, they were pretty well correlated with hepatotoxicity, either preclinically or in humans. It's a pretty low compound requirement, and it's a much faster turnaround time than trying to do in vivo studies. And the really great thing in this system is that you have free uptake of the ASOs, whereas the 2D cell culture systems, you actually have to transfect your oligo in. So this, and the fact that we're using human cells, it really makes that an attractive model to try to reduce compound attrition due to cytotoxicity. Now, of course, this is still in the early stages, and we certainly need to do a lot more testing of ASOs to truly determine is this going to be a valuable model or not. But potentially, it could be used as an earlier screen to reduce the animal studies as well as attrition due to toxicity. So overall, the conclusions here, oligos as a class actually present pretty unique challenges to try to assess safety. So they sit somewhere in between a large molecule like a monoclonal antibody and a small molecule. It's attractive because you can target the specific proteins, but you may also still have unwanted off-target effects that may not be detected in our non-clinical studies. And unlike small molecules that can readily cross cell membranes and get in, the oligos require transfection into 2D cell culture models, so it's just a lot technically challenging to work with. The hybridization dependent effects are really depending on the target, and they may actually manifest as an exaggerated pharmacology. In this case, we're targeting a virus, and so not hitting any sort of host target proteins. Certainly, you can have differences of in expression between your preclinical species and human. It's still quite challenging to deal with some of these hybridization independent effects. They are very molecule specific and are most common for the toxicities that we see. And it's really not related to the target sequence, but somehow is affecting off targets there. So despite the fact that we make pretty heroic efforts around the bioinformatics, trying to test things in vitro, even looking at a number of mismatches, I think I showed the data that up to 4 mismatches before you could start having an off-target. So the assessment of these oligonucleotides really might require utilization of some of these non-traditional models to identify any toxicity and successfully get into the clinic. So of course, I just want to thank everybody at Oligos who's done all of this work, and thank you for your attention today.
Thank you, Dinah. I think we have time for one question. I did want to ask you myself, actually, about these hero models. Did you find that you could use that to replace some of your other animal models, or is it just specific to HBV?
Yeah. So, those models have been around for small molecules for years, and they've shown pretty good correlation for small molecules and DILI. So ideally, we wanted to get into, what we've been doing with a lot of these is screening in the rodent, essentially. In this case, I think we could have reduced the number of animal studies quite a bit and really used this sort of system to rank order compounds and only advance the ones that were clean going forward.
Okay. Thank you very much.